So now we're going to have Dr. Brian Beal talk about his recent discoveries.
Okay, great. Thanks, Sarah. So I've left the spatial out of the discussion. There wasn't enough time. So I'm going to look at how soft-shell clams and green crabs vary in their recruitment through time in a place in the in the Harraseeket River. Now, this is a slide I show basically every time I give a talk, but I know that there's some of you that haven't seen this slide before. It's how clams get to be clams. They get to be clams because adults spawn down the lower left-hand corner. They release their eggs and sperm into the water column. Those eggs and sperm combine to form a zygote, which then develops into a trophophore. That trophophore swims for a day or so before it develops a shell. And then it has a shell stage, the veliger stage, for about 10 to 12 days before it begins to grow a foot. That foot then is used actively to allow that clam to, quote, decide whether or not it should settle. If it settles on something hard like this desk, it'll pick up and move its swimming organ and pull that out and go swimming away. Eventually, its biological clock ends in such a way so that it changes its shape, called metamorphosis, and then it settles to the bottom and we find a juvenile clam. So the question that I'm asking in the study that I did in 2017 in the Harraseeket River is, well, when do clams spawn and what does spawning look like? And I figured that there's probably at least 4 different scenarios that could explain clam spawning. So always the axis, I'm going to call this the x-axis right here, is time. So clam spawning could be really quick and short, so I call it short and intense. Or it could be broad and protracted, and this is what we think might be happening. Or it could be what I'm going to call skewed. It happens really quickly and it's really intense. Or it could be skewed, but it's a little more protracted. So I'm looking at these 3 or 4 different things and trying to determine what really is happening in this population. We know from the work that we've done at DEI and the Beals Island Regional Shellfish Hatchery that dates all the way back to 1987 is that you can almost set your clock by, if it's 50 degrees, clams are spawning. So that's what we found in the hatchery, and that's when we go out and we grab clams from the intertidal area and we say, well, is it 50 degrees? Well, yes, it is. So we ought to be able to get these clams to begin to spawn. So that we've used this tool for over 30 years, and it's been pretty good. And when clams are spawning, they have this very, very beautiful creamy colored texture, and then after they've spawned, they lose all that, and they don't look as good. They taste fine, but they don't look as good, and they're not filled as much. So if you're shucking clams, you know, you're going to get probably 1.5 gallons, almost 2 gallons, shucking clams in the springtime, and you'll only get 1 gallon or less from a bushel of clams in August, let's say. Now, again, we've talked about this before in these sessions, and basically for most of the towns out there, your clams are in the water column. They're swimming for 2 or 3 weeks, and really it depends on where your clams, where you're located along the coast. But essentially the clams are coming from the east if you're looking out from your town. I call this the salt shaker effect, where the clams are settling from the water column and reaching the bottom of the benthos. And recruitment, all right, so the word settlement is an instantaneous process, but the word recruitment requires some time. So when someone says, oh, I found 100 recruits, or we're going to sample recruits today, that's different than how many animals actually settled, because recruitment is settlement, but it's also some period of time after settlement, because we're not going to sample immediately after a clam settles. It's almost impossible. So we look at time, the animals are swimming, they're in the plankton, they settle, and then they go to the benthos, and they're called a recruit after some period of time. So depending upon when we sample, the recruits could be very small or the recruits could be very large. It just depends on that time. So the word recruit just is a clam sometime after settlement that's been lucky enough to survive. It's gone through this post-settlement phase. So there's some mortality for a lot of animals before we even see them as recruits. What could cause that post-settlement mortality? A whole bunch of things. Predation, disease, dissolution, competition, suffocation, enteromorpha, you name it. There's a whole variety of factors that can combine to kill clams. Okay, so in 2015, as Sarah said, we started using these recruitment boxes as a result of me using pet screening over plant pots in the Fore River and in the Wells River, or the Webhannet River. And I noticed that when I put the pet screen on my plant pots versus a larger screening, that, geez, I put 12 clams, as Kyle suggested, in these plant pots, and sometimes I get 230 clams back. The 12 hatchery clams plus, hundreds of wild seeds. So I said, geez, can we put this pet screening on these wooden boxes and just put them out on the mud and see what happens? And lo and behold, you've heard many times today what the results are. This is just taking the mud out, sieving it through a 1 millimeter mesh. It takes a long time to do, and it's well worth it because it gives us a lot of information, as Sarah said, about the productivity of an area that otherwise folks would say this is dead mud. So one of the things that we notice, or that you should notice, and I'm going to point out on this slide, is that we get a whole variety of different sizes of clams. We get clams that are big, we get clams that are small. The big one's about 1.25 inches. The smallest one is less than 1/8 of an inch. So I'm interested in why we have so many different sizes of clams. And so that was really the question. And the question was something of a conversation that Kyle and I had while we were processing these. And I said, geez, it must be that clams are spawning, over a long period of time over the summer. Otherwise, how are we going to get these small clams with the big clams? And he said, well, geez, could it be that they all settle at one time, but what we're seeing is differential growth, which means that some of the clams are growing faster than the other ones. So I said, gee, that's a great hypothesis, and so let's test it. So the questions are, when do clams, and in terms of their predators, the most important one, the green crab, when do they recruit to the flats? And what's the pattern look like? Is it, short and intense, skewed, etc.? And then what sizes of clams and green crabs actually survive into the fall and into the winter? So we set up a study in the Harraseeket River in an area where we had 2 years before placed these boxes, and we knew that this was an area of fairly high recruitment. It had been in 2015, it had been in 2016, we don't know why. The area that you see there, the 1, 2, 3, 4, 5, I'm going to call those blocks. And each block is about the size of this room. Okay? It's about maybe 30 by 50 or something like that, so it's fairly large. And there's about 250 feet from one block to another. And within a block, what we did was to place these boxes. 2 of the boxes look like this, which is just pet screen on the top and pet screen on the bottom. There's another box going around where the bottom is pet screen, but the top is 1/8 of an inch. I'm using 3.2 because that's how I'm thinking. It's 3.2 millimeters or 1/8 of an inch. So there's the large screen boxes and the small screen boxes. And every 2 weeks, starting on the 5th of May, 4 boxes were put into each one of the blocks. So that is to say 20 boxes were put out every 2 weeks from the 5th of May all the way to, well, here's time. Here's what happens in time. There's June, and then there's the July boxes, and then there's the August boxes, and then finally we get up to September 21st. So we put boxes down 11 times from the 5th of May to the 21st of September. And when I say we, that's the royal we, because it was Sarah and Chad Coffin that did 99% of putting out the boxes, and I thank them. That's 11 deployment dates. 11 times 5 times 4, that's 220 boxes. So that's what the study is talking about. In addition to putting the boxes out, every time we put boxes out, we took core samples. And these core samples were taken just exactly how Kyle explained for the number 10 core can. This is going to take about 10 pounds of mud. It's an area of about 0.2 square feet. And what we're trying to do here is to get a handle on natural recruitment on the mud flat in an area that's adjacent to these recruitment boxes. So what we're going to do then is to take the mud, put it into a labeled bag. We're going to come back to the Harraseeket Lunch and Lobster Float, the Coffin Wharf in Freeport, and we're going to sieve these samples on a 1 millimeter mesh. And again, we're going to do this on every one of those 12 or 11 dates. And this is what we found, which is not a lot of clams, but the pattern that we see here is really important. And the pattern is that we're seeing clams in the first 3 sampling dates, 5th of May, 20th of May, the 1st of June, and we're getting fairly high results or high densities, anywhere between 2 to 6 per square foot. And then all of a sudden, the clams start to disappear. And by essentially the end of June, all the way to the 9th of November, we see fewer and fewer and fewer clams. In fact, on the 20th of October and the 9th of November, we didn't find any clams in any of our samples. And every time we took 25 cores. So this was a lot of work, and Sarah did almost all the sieving. At the same time, we had this temperature recorder sitting on the flat, and what I did was I went back and I looked at an hour on either side of high tide and recorded in this particular representation here the temperature. So in May it started out around 48 degrees, the highest that we saw, which was fairly cool for this particular this summer. It only got up to 68. We've seen temperatures much higher than that, and then it was 50 degrees when we took the boxes up on the 9th of November. So what I'm going to do is superimpose seawater temperature on that rate of decline of these clams, and you can see that around 54 degrees to around 59 degrees is when we're seeing the greatest mortality of clams. That is to say, these are again from those core samples. All right, and this just gives us an idea of the sizes of these clams. And again, what we're looking at here is this axis is the size of the clams. This means that the clams are 1/4 of an inch. This means that the clams are 1/2 an inch. And this is how many that we found. So there's not a lot of difference between the first sampling in May and the second sampling in May. You can see that the clams are growing a little bit, but there's not a lot of small clams. Then all of a sudden on that June 1st sampling date, we start to see small clams showing up. And then those small clams begin to disappear. And we just continue and continue and continue and continue all the way to the 21st of September through the 4th of October. And you notice that through time, we've lost all the clams that were there in May. And just a few small clams exist. In fact, we're getting small clams in September and August and October. Where did they come from? So this is kind of a prelude or precursor of what we're going to see, what I'm going to show you in the boxes. Because it turns out that the sampling that we've done in these cores is pretty predictive of what we're going to see in the boxes. It's pretty interesting. So to summarize the core sampling, what we've got then is that they were taken, those samples taken prior to June, had soft-shell clams in them that had settled in 2016 and overwintered successfully. Those sizes range from about 1/4 of an inch to 3/4 of an inch. Recruits from this year started showing up in the 1st of June core samples and ranged in size from about 1/8 of an inch to 1/4 of an inch. After seawater temperatures reached 54 degrees, we saw that marked decline in clams and their abundance. And then that decline continued until the last 2 samples were taken, which contained no live clams at all. Recruitment occurred throughout mid-August and perhaps into September. And then spawning, I think, appears to be protracted from what we found from the core samples, occurring from about mid-May until at least early to mid-August. Okay, so that's the story from the cores. Now let's see what the boxes give us. So this is a picture of us taking the boxes out in November, and it's curious. Some of the boxes might weigh 10 pounds, some of the boxes might weigh 40 pounds, and that weight depends on when we put them out. So the boxes that were put out later on in the year don't have the amount of mud in them that settled from the water column. So here's the deal. Does that look familiar? Well, it should, because what it's showing us is that there's really no difference in the number of clams that when we put the boxes out on the 5th of May or the 20th of May or the 1st of June. So that tells us that clams were not settling in May that, at great rates. It looks like the settlement is occurring sometime between the 1st of June and that 15th of June. And so what we're looking at really then is 2 time frames where we get just a bejesus amount of clams settling. And what's curious is that, whether or not it was the Searsport group this morning, or whether it was Bailey, or even if it was Kyle with protected nets, we're averaging about 200 clams per box, which is about 100 clams per square foot. We've heard that now 3 times today, about 100 clams per square foot. But we're also finding clams that are in the boxes as late as the 21st of September. So what does that mean? Well, first of all, even though Sarah recommended pet screening, it turned out that the flexible, the larger nets actually gave us about 77% more recruits. But I would agree with Sarah, don't use that 3.5 millimeter stuff. It's very, very easy to rip. And 25% of our boxes were actually, I had to throw the data away because the boxes had large gaps in them. And guess what was inside those boxes with large rips in them? But take a look at the difference that we see between the 1/8-inch netting and the pet screen, the 1/8-inch netting and the pet screen, the 1/8-inch netting and the pet screen. It's pretty clear that the pet screen isn't really doing as well as the 3.2. Let's superimpose temperature on top of that, and honestly, it looks almost identical to the core samples. It's pretty wild. In other words, when water temperature is about 54 degrees, that's when the bulk of settlement looks like it's occurring. So what then do we use to describe settlement, or recruitment, I should say? And so recruitment looks like it's skewed, but it's protracted. We're getting a lot of clams settling in a short amount of time, and then there's protracted settlement all the way up to September. So if recruitment is skewed and protracted, then what should those sizes of the clams be in the boxes at the end of November? And so the answer, it turns out, Kyle, is that it depends on when the boxes were deployed. And so those boxes that were deployed, early on, the 5th of May through the 1st of June, those should have 2 distinct sizes of clams in them. They should have clams that settled early and grew big, and because they were out there as late as September, they ought to have some small clams in them as well. So the boxes that go out early will have the largest variation in sizes of clams. So those deployed after June should have progressively smaller and smaller clams. In other words, if you put them out in early May and June, you ought to have a lot of small clams, but some very large ones as well. If you put them out in early to mid-August, you have probably an equal amount of large and small, or at least that was the idea or the hypothesis. And if you put them out in late August or early September, you'll have a lot of small clams and just a few, maybe not that many, large clams. Well, here's what we saw. If we put the boxes out early, we have just a little blip of small clams and a lot of large clams. These are 25 millimeters, as I say, is an inch. So we were finding clams as large as 1.5 inches in some of those boxes. That's pretty incredible. That's a fantastic growth rate. There weren't any clams in those boxes to begin with. So to get a 38 to 40 millimeter clam in one growing season is really wild. So that kind of tells us when we put those boxes out in the springtime what we're looking at, what's going on. No, I can't use those guys. All right. So this is what we get when we deploy them in May. This is the size distribution when we deploy them in July to mid-August. We get a lot of small clams and we get some large clams, but we certainly don't see any that are 1.5 inches or 1.25 inches. If we put them out very, very late in the year, August through September, we're still getting clams, and that's really important if you're a clam digger. This slide right here says a lot about your future. Small clams. Lots of small clams. So this is the average size of those clams through time. And as you can see, the earlier those boxes are put out, the larger the clams and on and on. So this is what relatively the size of the clams are through time in those boxes. So to summarize the boxes for clams, recruitment in the boxes essentially mirrored the results from the cores. We had a really good peak in clam recruitment that occurred sometime, we think, between the 1st and the 15th of June, when the seawater temperatures were between 54 and 60 degrees Fahrenheit. Recruitment was skewed and protracted. It occurred in all boxes, deployed even as late as the 21st of September. Clams recruiting to the flats in early summer are exposed to intense predation and most die. That bulk of clams that settled, they didn't make it. They weren't in the cores. How many clams did we have in the cores in October and November? None. So where do those clams go? They go where the Searsport students are going. They're going to get them. So they settle, but few of the clams actually make it. And it turns out that the ones that make it are the ones that settle the latest on those flats. And that's why when you go and you take core samples in the springtime, you find clams that are 6 millimeters, 8 millimeters, 10 millimeters. Where are the 38 millimeter clams? Where are the 1.5 inch clams? They're gone. They don't exist because they provided the energy for green crabs the previous summer. So the clams that survive to live through the fall and winter, they're typically less than 1/2 inch. And those are the ones that are going to become the commercial clams in the future. How about green crabs? Well, the boxes can't not disallow green crabs to come in, so we get green crabs in some of these boxes, which means that we have to take with a grain of salt the actual productivity that we're talking about. It's probably higher because we can't keep the green crabs out. So what do we know about green crabs? Well, we know that they release their eggs in May to June. The larvae are settling August and October. They can go into the winter at sizes that are 1 millimeter or smaller to maybe 10 millimeters. But they're planktonic. And so it's like the salt shaker of soft-shell clams. They're going to settle out of the plankton and onto the bottom. And when they settle as this thing called a megalope. It doesn't look anything like a crab. It has to do one more shed or molt before it changes its shape and actually becomes a juvenile. This is what they look like in the plankton. And this is what they look like when they settle to the bottom. And this is what they look like after they've shed and end up looking like a green crab. And that's the size that's going into the winter. So the green crabs that we found, this is the number per box, follow roughly this kind of skewed and protracted event here. I looked at the number of green crabs that were smaller than about 3/4 of an inch and crabs greater than 3/4 of an inch, and found that, especially during the early part of the summer, we were getting a lot of crabs that were in both of those size categories. In fact, if we look at the sizes of green crabs, I'm going to take the 5th of May and the 20th of May in the boxes. Now, again, these were when they were deployed, so how is it that we're able to get large crabs and small crabs in the same box? We get a large crab in the box because it crawled in May as something that was smaller than the mesh. And then it came in and it started feeding on whatever organisms were in here. Could have been worms, it could have been small clams, whatever. But it fed, and then it grew, it shed, and now it's trapped. So you put this box out in May, and you're going to get large crabs in November. Well, you're also going to get small ones because sometime in July or August or September, the crabs are going to come from the plankton and they're also going to settle on the mud, and you're going to get small crabs. So the boxes that get put out early have large crabs and small crabs in them. And you'll see as we go through July and now into September, the ones that were put out in September only have the small green crabs, but they're still there. So when we look at the sizes of these crabs through time, we can see on average what they look like as a function of the boxes that they were in. So it looks like the largest influx of the smallest crabs are occurring in August. That's when the settlement of that year's crabs looks like it's the most intense. So to summarize for the recruitment boxes for the crabs as small as 2 millimeters, or less than 1/8 of an inch, entered the boxes early, even in the smallest mesh. And some of these individuals grew to a size of 1.25 inches, 1.5 inches by the time that we sampled in November. The crabs occurred in boxes deployed on each of the 11 dates, so we couldn't keep them out. They were always going to be there. No crabs occurred in boxes with the smallest mesh on the 21st, but that was the only one. Recruitment appeared to begin sometime between the 10th and the 23rd of August, when the largest influx of small crabs occurred. And these crabs are likely to continue to settle to flats throughout October and perhaps early November. And then they overwinter at these really, really, really small sizes. So, the last sentence here is that survival success of 0-year class crabs over the winter is the key to the survival success of softshell clams the following summer. If we have intensely cold winters, like occurred between 1960 and 1965, you couldn't find a green crab in 1966, 67, 68, and 69. People looked for them. Walter Welch at the Department of Marine Resources published a paper in 1968 on green crabs in Maine. Couldn't find any. Will this winter's cold that we've got in December and January, will that be like 1962 or 63? My prediction is no, because the water temperature was warm at the time when it was so damn cold. If the ice that we saw had occurred later in the winter, maybe, but we'll see. I'm hoping that I'm wrong. So I've got 2 wrap-up slides, and then I'm done. This is my chance to be an artist. Now, I can't even draw a straight line. So here we go. This is what I see. So we have a circle that represents the year. Springtime, summertime, fall time, and winter time. And this is when clams are spawning, according to the recruitment data that I got. Okay? So they start spawning let's say sometime around the 15th of May, and they can be spawning all the way up until maybe the 20th of August. We get an intense settlement of soft-shell clams sometime between the 1st and the 15th of June. And then we get more, but we get a protracted recruitment all the way up until around the 21st of September. But we don't get as many. And then some of these clams, a few end up surviving the fall and into the winter. And there's a little bit of growth all the way up until around November, when the clams stop growing, and they'll be the same size from November until the following late March. And then beginning in late March and early April, the clams will begin to grow. And it turns out that, at least in this particular study site, that you could actually get a legal-sized clam 1.25 years after settlement. So sometime in late July or early August of the next year, you can count on, if they survive, legal-sized clams. When do you find green crabs? This is going to get really messy, and I'm sorry, but anyway, you find green crab. Huh? That's good, because I've got 15 more slides. No. So this is when you find green crabs that are ovigerous, that have eggs. And this is when those megalopes are beginning to settle. Okay? And you'll find these megalopes, these 1 millimeter crabs, you'll find them continuing all the way into spring, and some of those are probably going to end up in these boxes. However, some of these crabs metamorphose, and you have small 5 millimeter crabs going into the winter and into the spring. And some of those that are 1.5 millimeters will enter into these boxes. They'll begin to grow, and then they'll just raise havoc on anything that's settling. And that's what happens to this intense settlement peak. We lose those clams to those predators. So 11 months after settlement, we have 1.5 inch green crab. Okay, so this slide is with malice towards none. And, here's what I think. Previous work has shown that it really isn't possible to set and haul enough traps to make a dent in green crab populations. And unless we have a string, not one, but a string of very cold weather, or there's some kind of disease that wipes out green crabs. They've been here since 1905, so they haven't seen a disease yet that will wipe them out. But maybe. But if none of that happens, then green crabs are here to stay, and we know that seawater temperatures are increasing in the Gulf of Maine. So that means that green crabs are just going to continue to increase. At 200 clams per box, clams are recruiting at a rate of 100 per square foot. We saw that in 3 different places. But the core samples are telling us what's really happening in the wild. And that is that the majority of clams die. And previous studies have shown that most are consumed by these green crabs and other predators, sand crabs and milky ribbon worms and even fish. You open up a mummy chog in August and take a look at its gut. It's got small clams in its gut. They feed on clams. Moon snails. So that is 100 per square foot isn't enough for both humans and non-human predators. It may be possible to increase recruitment rates if there were more larval clams settling from the water column, but how do we do this? So I have 2 suggestions on how we increase the number of clams settling to flats to swamp out predators. One method would be to stagger closures of the fishery by region along the coast for 2 to 3 weeks to allow more clams to spawn each year before they're harvested. We can call it a spawning closure. And then my favorite, and this is where Cole and I differ in the legislature, one of the places that we differ. Another method would be to allow clams that are larger than 3.5 inches to spawn. Just leave them. using the same logic that lobster fishermen leave 5-inch carapace length lobsters in the water, the same logic that sea urchiners leave 3.5-inch sea urchins in the water, because they're spawners for life. So it turns out that soft-shell clams also have an increased number of eggs as they increase in size. So I call that the maximum size. And I'm done. There you go.